Bismillahirrahmanirrahim and welcome back to this next video, and this is the 15th video in the series of videos on immunology, and this is the part 2 on the pathogen recognition. In the first part on the pathogen recognition, I told you that an effective immune response that relies heavily on the nature of the invading pathogen and the cells and the molecules of the immune systems that are activated, that depends on the chemical structures present on the pathogen and the location of the immune response in the host. And I've told you that different chemical structures mean different kind of the pathogens and the microenvironmental cues that are needed to be detected for an effective and specific response strategy. Then I've told you that the process of pathogen recognition that involved an interaction between the pathogen and the recognition molecules that are expressed by the host cells. So if you talk about these recognition molecule of the host, I've told you that these recognition molecules are, like in majority of the cases, they are membrane bound receptors, but there can be soluble receptors or secreted recognition molecules. When you talk about the pathogen, so a variety of the things from the pathogen that are recognized by these recognition molecules, and that include the whole pathogen. It can be the antigenic fragments of the pathogen, or they can be the secreted products of the pathogen. So the interaction between the ligand and the receptor, I've told you, that results in a cascade of the events that ultimately lead to the labeling and destruction of the pathogen, and this immune response that involve both the cellular immunity and the humoral immunity, which are the two arms of the immunity. In this particular video, I want to focus on the intracellular pathogen, because I've told you that the immune response that is dependent on the nature of the pathogen. So the nature of the immune response that will vary depending on the type of the pathogen involved. And in this particular video, I want to focus on those particular pathogens which are intracellular. By that I mean that these pathogens, they are going to move inside the host cells, thereby causing the diseased condition in the host. When you talk about these intracellular pathogens, the most appropriate example that would be the viruses. These viruses, they are tiny. Why I call them tiny, that will be clear in a while. So these viruses, they are tiny, obligate intracellular pathogens, and they spend majority of their life cycle residing inside the host cells, and thereby we call them is the intracellular, that means within the cell. This is just an image to give you the idea about the sizes of the viruses. So if you talk about the most common one, so let me start with the hepatitis C virus. If you look at the size, this is 50 nanometer. The hepatitis B virus, that is only 42 nanometer. If you talk about the rotavirus, this is only 80 nanometer. If you talk about the influenza virus, the one that is disturbing our life every changing season, so the size is only 100 nanometer. If you talk about the SARS, one of the thing that we have seen in the current era from this particular family, that was COVID-19, and look at the size, like 120 nanometer. The herpes virus, that's only 200 nanometer. What I mean to say is that these are tiny viruses that is creating, or that is infecting the host cells, creating a lot of infections in the host. This is just to give you an example how these viruses, they enter into the cell and how they become the intracellular pathogens. So this one is the most common virus, the influenza virus. What this influenza virus do is that it is going to attach to a target epithelial cell, and the virus is going to enter into the cell by the process of the endocytosis. And once the virus that enters into the cell, the epithelial cell in this particular case, they are going to release their content, and the viral RNA that is going to enter into the nucleus, which is going to make the copy of its viral RNA using the viral RNA polymerase. So when the viruses, they make the copies of their genetic material, they are going to make viral messenger RNAs. These messenger RNA are going to move into the cytoplasm of the cell, and they are going to make the viral proteins over there. So new viral particles, they are going to be made because they have already replicated their genetic material, they have made their proteins. So new viral particles, they are going to be made, and they are going to be released into the extracellular fluid, and they are going to infect the fresh epithelial cells. So the cells which are not killed in the process by the immune system, those particular epithelial cells which are not killed by the immune system, they continue to grow and they make the new viruses, thereby complicating the process. So this is just a simple example of how these viruses that enter into the host cell, thereby making them the intracellular pathogens. So an effective defense is they are residing inside the cell. So an effective defense strategy must therefore involve the identification of the infected host cells, because there are a variety of the cells. So you need to have a mechanism that can identify the infected cells. By that I mean the cells which have been infected by the virus. Now this means that there should be some immune cells that are capable of detecting the changes that occur in a host cell after it becomes infected. And this is achieved by a range of the cytotoxic T cell, by a range of the cytotoxic cells. We will have a detailed discussion on the cytotoxic cells, but for this particular time, just keep in mind that a variety of the cytotoxic cells, they can identify these infected cells. But one of the most important of these cytotoxic cells that is known as the cytotoxic T lymphocytes. We call them as the CTLs, the cytotoxic T lymphocytes, or they can also be called as the cytotoxic T lymphocytes. And if we are talking about the cells, that means that the cytotoxic T cell, they are going to be part of the cellular immunity, not the humoral immunity. So what these T cells, they are going to do is that in this particular case, the recognition molecules, as I've told you in my previous video, that the host are going to produce recognition molecules. That could be the membrane bound receptors, that could be soluble molecules. So in this particular case, in case of the intracellular pathogens, the recognition molecule that must be present inside the cells so that they can interact with the, you can say, ligands produced by the pathogens. Now these intracellular receptors, they are going to bind to the viral protein. As I've told you that these viruses are going to make their proteins. So these intracellular receptors, they are going to bind to these viral proteins present in the cytosol of the host cells. And this interaction is actually initiating an early warning system, altering the cells, the neighboring cells, the presence of an invader, in this particular case, a virus. Now, the most, you can say, important strategy or the most appropriate strategy in the intracellular pathogen is the sacrifice of the virally infected cells. And that, you can say, becomes the only way to truly eradicate this type of the pathogen. Now, in general, this sacrifice is very good for the whole organism, because if you are not going to kill these cells, they are going to harbor these viruses, and the viruses are going to increase their number, and theoretically the infection is going to be there throughout your life. So the good thing is to clear these virally infected cells from the body. So in majority of the cases, this sacrifice is very good for the whole of the organism, although there are instances where this sacrifice can cause disruption to the normal function of the immune system. For example, if you talk about the human immunodeficiency virus, the HIV virus, the HIV is going to infect a specialized type of the T cells, which is called is the T helper cells. And we call this as the T helper cells, because these cells are going to guide the behavior of the other immune cells. I'll show you an image at the end of this video. So these helper cells, they are going to guide the behavior of the other immune cells, including the B cells, and are therefore very important for selecting the appropriate immune response. So if too many of these helper cells, they are destroyed or they are rendered non-functional, many of the directional cues needed for a healthy immune response, for an appropriate immune response, they are going to be missing, and fighting all types of infection become problematic, because you are going to be missing with an important component of the immunity. Now the resulting immunodeficiency, why I call this as the immunodeficiency, because you are going to have a deficiency of the T helper cells. That means they will not be able to guide the B cells what to do. They are not going to activate the B cells. Therefore we say that there is going to be deficiency of the immune system, and this immunodeficiency allows the opportunistic infections to take hold and potentially kill the patient. Now, if I give you like the, if you talk about these, this is the repetition of this particular slide. Forget about this. We already discussed this thing. This one is, you can say, a macrophage, and this is a helper T cell. So the helper T cells is going to help the macrophage clear the phagocyzed pathogens from the body. If you talk about this helper T cells, that is going to help the B cells in clearing of the foreign pathogen. So imagine if there is a deficiency of the helper T cells, what are you going to do? The end result would be an immunodeficiency, and the opportunistic pathogens, they are going to thrive in the body, thereby creating complication for that particular patient and have the potential to kill that particular patient. So if you like the video, please subscribe to my channel, hit the notification bell, comment on the video if you have some comments, and do share it with your friends. I'll see you in the next video.